Okay, so I am excited to introduce Sofia Sanchez. I actually just randomly came across her blog post on Medium, and I love this. Please build robots to automate plant tissue culture. And I was like, this is perfect for Open Auto Club. I think one of the challenges in automation is often, you're an automation practitioner and you just want to build stuff, so you get really focused on how you're building. And I think it's also really useful to think about like what would be really useful to build. And I really liked this blog post, so I asked Sophia to talk about it, and she agreed, which is great. So I will now hand it over to Sophia. Please welcome Sofia Sanchez. Let's give her a little welcome.
Yeah, thank you. Happy to be here. So, yeah, quite a random find, but I'll tell you a little bit more about, I guess first, you can't see my screen, can you? Oh yeah, you can see it now. I think
Yes.
so. Yeah, cool. So, my background is in bioengineering and like plant synthetic biology. So, how do we create new crops or engineering tools for plants? And I'll talk a little bit about like what areas interest me in that space. The first time that I ever came across this field was actually people who store data into DNA and then into plants. They are called Grow Your Own Cloud. I've been in iGEM, in biomaterials, and in all sorts of weird explorations, also a little bit, mainly in cotton. So generating new cotton varieties with different properties. I'm based in Mexico, and so yeah, that's a bit about me. Then about the talk, I will focus on why grow plants in jars first, like what is this plant tissue culture thing? And then, oh yeah, yeah, I just saw the chat. And then, how do we actually make these plants in jars? What other people have done in the past, and then we'll go over some questions. So yeah, like we know about like the drought difficulties, the increasing pests, the climate change stuff. And those are, well, I guess I see plant tissue culture as the base for generating these new plants. So whenever we are trying to either genetically engineer a crop or select for a different crop or just study it, even for basic research, like understanding how different tissues develop within a plant, plant tissue culture is like this way of isolating these plants and well studying them in a more, let's say, in a different environment, in a lab environment, pretty similar to what we do in biomedical research. And I wanted to play this video that sort of goes into uh, how we would create trees that capture more CO2. So,
Sophia, should there be a video on the screen right now? We're on slide 2
oh, there
currently.
should. Okay, let me share on my screen. Great,
Okay, now we see a video.
great. So, where do I share like the sound, the audio?
That is a good question. I think it's in
It
the
looks like
share
you're
button.
muted just on your own video playing.
You
Like
can
next
see
to
at
the
the
play button, it's
bottom
muted.
left. Yeah, the bottom left there seems to be
Yeah,
a
but I don't
sound.
know if it'll be
If you
better. Okay,
can you click on it, maybe?
good. Can you listen to that video now?
It's not playing for me.
Okay, let's
You're
go.
working
Yeah,
in science, you need to use whatever materials you can get a hold of. That's Maddie Hall. Hello. CEO of the company that's engineering these trees. So this is where we do a lot of our actual genetic engineering. So where we're incorporating new genes that
So I'll pause there because these are some different tissues of plants that we work with for genetic engineering. They're called callus or calli, and so yeah, just to like get a sense of you have the plantlets, which are like the ones we normally know. It's just like a mini plant in vitro, but we also have different types of tissues. These are the callus are sort of plant stem cells.
come from other plants. This is where all of our plants start. I'll show one example right here, and they've been put
So I'm
on
actually not seeing the video move.
Oh, you're not seeing it move.
Yeah.
No, yeah, we're at,
I noticed in the bottom right, it says 4K. Maybe that's too much data. So maybe in
Oh,
the
okay.
if you drop that down to something manageable,
Yeah, let's see.
you should be able to click on that real
And that
icon that
allows
says
for the
4K.
Could you see it
maybe
now?
stop sharing and then reshare.
Yeah, let's see. This will be important because a lot of the like actual tissue culture I have like YouTube videos to show you guys. So let's see.
As
Uh,
you're resharing, there should have been checkboxes on the right for share sound and optimize for video. And optimize for video is probably the one it needs.
hmm. Okay, let's see. I see. Thanks. How about now?
Plant cells that have had the new genes
Yes,
incorporated
works.
to
Okay, I'll just replay this.
creative science. You need to use whatever materials you can get a hold of.
That's Maddie Hall,
Hello.
CEO of the company that's engineering these trees.
So, this is where we do a lot of our actual genetic engineering. So, where we're incorporating new genes that come from other plants. This is where all of our plants start. I'll show one example right here, and they've been put on what's called selection media, and that allows for the plant cells that have had the new genes incorporated to continue to grow and eventually start to make shoots and roots.
All right, here's how these trees are different from their unaltered friends. So you already
Well, that was just, a glimpse into how a small part of the lab looks like, and what they're doing is genetically engineering the trees to optimize like their carbon capture abilities. And you could see like the different the ones that were in the petri dish were these plant stem cells, the callus, and then they can genetically engineer those and then regenerate those into a whole plant that can actually serve the functions they want. So and create, I guess, new quirky functions for plants like glowing in the dark, which is something that I'm interested in as well. We did it, yeah. Now, what is it really? I would like to, let's say, summarize the process of plant tissue culture into 4 stages, and specifically the ones that I see having the, let's say, more opportunity for automation are establishment and subculturing, and acclimatization. I would say that's like when you're transferring these plants from in vitro to ex vitro to actually the soil and the fields, which is also, I guess, a repetitive process. So I'll go through some videos now, but overall, you first need to take a tissue. You can, depending on what you want to do, you might select a shoot, an axillary bud, which is kind of you can have like nodes which are between the leaves and the stems of the plant. You can even have roots, you can have leaves, but usually axillary buds, like these nodes are, or at least those are the ones that I've seen people working with the most and the ones that I've worked the most with. You of course need to make media, another repetitive process, right? Sterilize the explant, which I'll start by saying that it's something that I haven't seen others automate that much, but if you're taking lots of these explants from the fields from another plant and you want to bring them in vitro, you need to go through some sterilization first, and that can be hectic as well. And well, then plant into the media. Then from this first establishment, it doesn't need to be just one jar, but let's say you're at a very low quantity of explants and of jars, then you grow those. It takes between 2 weeks to 1 month, a little bit more depending on how fast the plant grows, the variety and the species that you're working with. And then you can, cut those into lots of more new explants. So I guess a property that a lot of plants have is that you can, let's say, regenerate those as long as you cut it, it can grow into another explant of its own. And this can actually be done in bioreactors, which for plant tissue culture, they're not super specialized. People even use or reuse water bottles, and they can just create a very simple aeration system with some pumps, and this is mainly for also like practicality purposes. But it's mainly the same process, right? Picking the explant from the previous jar, cutting it, then putting it again into new media. And then since you don't have this media doesn't let the plant grow roots. Mm, you then want to, if you want to regenerate the plant, you'll have to put it into yet another growth media that does allow it to grow roots, so you can then transplant it into the trays. I don't know if, this is more or less clear, if there's anything to clarify here. Any questions so far?
I just have one question. I'm kind of getting caught on the pickaxe plant from, what is that last sort of thing? It's like a one with two lines around it.
Which step? Oh, yeah,
Oh,
right. From step one. Yeah.
okay, got it.
Yeah.
So people really just like, you're talking about like just an old water bottle, like you can put your plant in there and just kind of, and there's no issues with contamination or anything.
Yeah, right. So when you're propagating these plants and you're doing all the cutting and so on, you do it inside a safety cabinet, and as long as you do proper sterile technique, meaning you rub your hands with lots of alcohol, and sometimes I'll go through it in the videos. A very weird thing is that people tend to use alcohol lamps to sterilize also the instruments that they're working with. So yeah, that's how you prevent contamination. But as for the water bottles, yeah, I suppose also making sure those are sterile. There wouldn't be any problem.
I guess you can use techniques they used in like the homebrew world to sanitize the bottles.
Yeah, what I'm curious here, well, I guess they must use like new water bottles. There are people who also use, well, um, glass bioreactors, or glass bottles, so they can also reuse them and put them into the autoclave, because I guess that wouldn't be the case for these plastic ones. And yeah, so I'll move on to what is the most labor intensive process here. And I mentioned it in the article. It's mostly for the developed world, in which, labor costs are higher, that this actually becomes a problem. Because, you in like even in countries like Mexico, you can train anyone to do this tissue culture, and it's not very complicated. But in, the Netherlands tends to be, one of these countries that's leading, or that has many of these companies that do this type of work. Labor is higher, and these mostly repetitive processes are the ones that would need to be automated. I see something in the chat. So could these bottles be put in a pressure cooker for home? I don't think those bottles could. I think there's something called the biocoupler that some people created that's like smaller, but yeah, it's glass. So I guess that one can be autoclaved. I'll now, oops, the videos. So let's see. And here.
What if your travel buddy was the most functional?
Now it is time to start subculturing.
What if your travel buddy was the most functional bag ever? Being organized.
Can you see that?
Yep.
Yeah.
Great, thanks.
Here, I will be splitting up the plants and removing any dead plant material and roots.
Like making salad.
And now that the plants are separated, I can add them to the media.
Are there any antimicrobial agents in there in the media?
There can be, and that actually helps a lot. They tend to be expensive. Can't remember the specific cost right now. But yeah, people tend to use them sometimes. Also, active carbon sometimes can prevent or help prevent contamination. But usually, and as I've worked with cotton, as long as again you're having proper sterile technique in the safety cabinet, there should be no need for antimicrobial agents. And fungus tends to be actually the like main issue here, not really bacteria or anything else.
Yeah.
And also this does look like a salad and like a very, I guess, bushy plant, but it tends to change. Some explants are different and a little easier to work with. You don't have to remove so much like brown leaves and so on.
This is how easy it is to change the media in your biocouplers. This is the perfect opportunity to split your plants if needed or remove any dead plant material. This is how you sterilize a plant for tissue culture. I'm using a begonia, so I'll start by cutting off the leaves and cutting around the nodes. Make sure to sterilize your tool before cutting your plant to prevent the spread of diseases.
So people can either sterilize their tools by using some glass beads, like here. I think that's what she has used. You can also, of course, use like alcohol, 96%, or the alcohol lamps that I was talking about, just like putting the thing on fire for some time. And I don't know why she explained this process after she did the like transplanting, but this should, like as I explained before, be done in the initial part of the process. And this is something that I haven't seen be automated yet.
Now I'll make my 10% bleach solution where I will use 10 milliliters of bleach and 90 milliliters of water. And now I'll add 1 drop of polysorbate 20. Now I'll add my plants to the solution and I will shake the jar for 15 minutes. After the 15 minutes are up, I'll pour out my bleach solution and rinse with sterilized distilled water. Now we rinse 3 times, 5 minutes each. Now that the third rinse is done, I'm going to cut the ends of the plants before I add to the media.
Have you ever considered at-home needling?
All the time.
Something to mention here is how she's also using some tubes. So sometimes people, depending on the price of the flask and what explant they're working with, what type of plant, whether they want it to grow taller or shorter or whatever, they'll use either like these squared flasks or these tubes. I'll show you in a next slide.
Today I will show you how to acclimate tissue culture plants to soil. I chose this plant because there was a lot of growth in roots. The first step is to take the plant out of the container and take out any old media from the roots. Now let's take the plants to the sink to give them a rinse. Now let's rinse the plants and make sure to get rid of all of the gel. Now I'll add my soilless media, which is a combination of 50% moss and 50% perlite. It is essential to keep humidity high in the first few weeks using a humidity dome.
Yeah, so that's pretty much the whole process in a nutshell. And I don't know if you guys have any questions so far.
Hey, Sophia, I was wondering about the two parts of that biocoupler. Like, is the growth medium in the upper part and it's just humidity kind of stuff beneath it? Or how do those two up and down work together?
Right, so here they're working with liquid media, and the reason to have the two parts is because sometimes you want the plants to get like air to be like, yeah, sort of breathing in a way. And so you can turn them around. And when you, yeah, on one side, they'll be like taking nutrients from the media, and on the other side, they'll be sort of breathing. So that's, yeah, the purpose of them. And this can also be like, I think they also created some sort of system to turn them around, which shouldn't be that complicated. So yeah.
Cool, thanks.
And then this is like my ask, like plant tissue culture. Well, here you can see like, so far in the videos you've seen people doing, let's say, low throughput, more or less, work. But the real opportunity, of course, here is for big ag companies that are making up to like 2,000 jars a day is, I think, the limit to what one single person can create or work with. And especially if you want to generate new varieties and like test many different experiments, or what most people, even in, like Mexico where I live, use this technique for is like generating pest-free varieties. Well, these are actually plants that are going to go into the fields, and we're talking about thousands and thousands of units. So, most commonly used instruments, as you could see in the videos, the tweezers, the scalpel, the petri dishes. Sometimes people used to cut, like put the explants there and cut them there. The safety cabinet, of course, and the different flasks that I talked about. The great also opportunity for automation here is that if you can have like a single sort of robotic arm inside one of these safety cabinets, is that you're saving well a lot of loss in contamination. So I guess that's also a great opportunity. Now, what other people have done, these are three main systems that I'd like to highlight and the ones that I was able to find so far. So let's look at them.
Smart clone platforms radically change tissue culture. Plantlet pods allow fast robotic movements and better growth through control of the plant microenvironment. The system boosts operator efficiency and best uses their key skills. Touchscreen commands allow simple control of the various modes available. Cloning is illustrated with grapevines. Input plants are in the tray on the left with agar-filled output pods on the right. The robot uses its pneumatic grippers to load the pod stand in front of the operator, 2 input pods on the left side and 2 output pods on the right side. The operator follows the red lights indicating where to take explants on the left and where to plant them on the right. The operator controls release and grading of pods using 2 foot pedals. Tips and nodes can go into separate boxes. Cloning typically averages 6 seconds per plantlet.
Which is, by the way faster than a human alone could do it, or at least an inexperienced one. And well, what I'll highlight so far from the video is that the system is only taking the trays and putting them in this, like, I don't know, support system they have, but the human is the one that still does the cutting. So it's like making the process faster, but not necessarily full automation.
Here we see potato plantlets being cloned, but with 2 explants put into each pod, which can be beneficial. The speed is good at an average 5.2 seconds per pod, only 2.6 seconds per plantlet. A database records the history of each plantlet over many generations. Teak have a similar form to potatoes. The large leaves must be removed, which is easily done without rushing, and the plants are again graded into tips and nodes in separate boxes, as these have different growth behaviours. Strawberries grow differently to previous examples. They require a basal dissection through the agar rather than aerial tips and nodes. In this case, the input plantlets are in conventional pods, but the dissected explants are transferred to a basket pod of different design. The pod stands are easily swapped when a clean one is needed. Locking pod stands are available to better secure the pods, such as for basal dissections. The locking catch can be seen closing over side pins on the pods. Here we see hemp being dissected using the new Gemini with twin grippers. They pick up 2 pods at once, doubling output. The speed is reduced to 15%, so the operation can be seen clearly. At full speed, a plantlet can be processed every 3 seconds, which is 8,000 per shift, or 2 million per year, and over 8 million if operated 24/7. Another operation is fast pod transfer between any sets of boxes to change media. These take less than 2 seconds per plantlet without operator involvement. Pod fingers and lid horns are automatically cleaned and sterilized with up to 6 bars of ethanol and other solutions. Trays are easily removed and the box is freed with quick release catches. It's so versatile it can even stir your coffee for you. SmartClone is a powerful system suitable for most tissue culture applications. It can substantially improve the throughput of operators and facilities. SmartClone systems allow more efficient production of better plants.
Yeah, what I highlighted here also is that I don't know how much the shape and size of the trays would need to be adjusted to work with different explants. So again, for companies that work with a wide variety of plants, yeah, it can make the process faster, but it can also be limiting in that sense. And yeah, as you could also see at the end, the human still needs to like put the tray into the safety cabinet, which is something that other systems do automate, like this one I just found a couple of days ago.
We talk about plants, and they are individual. Each plant is different, and because we use AI, we can handle and process these plants. And I think this is a worldwide unique system at this point. This machine is a PTC lab on a few square meters. So in detail, this is a quarantined environment, and you put in cups with plants. The machine opens the plants, takes out the plants, analyzes the plants, cuts the plants, and put.
This one uses a laser to cut the plant instead of having the scalpel, and they say it can be better for the plants to they can better regenerate in that way. That's well the innovation that they highlight the most.
Puts the cut plants back into a new cup, seals the cup, and puts it out of the machine.
Plants are growing better if they are divided with laser in comparison to a manual scalpel cut, what we usually have. And we can see that the plant is recovering faster, growing better. So that's also our claim, smarter propagation, better plants, because we save on cultivation time and we have stronger plants.
The world needs more plants. There's going to be up to 10 billion people that have to be fed until 2050. How do we do that? We can only go by finding new technologies, having really innovation in place. So here we have robotics, we have AI, vision recognition, the plant science.
Of course, their computer vision system is also cool because that I would suppose they can train it on many different types of tissues. And, again, it's not only that you're working with different plant species, but also you might be working with a leaf, you might be working with an axillary bud, with a node, with the callus that I was showing you at the beginning.
So we're proud that RoboCut is made in Germany, but we are really proud that it is made by a highly international team.
And try it out and perfect it. And by making a machine or developing a machine that can produce plants that help provide oxygen, that help feed people, I think
that's where they stopped talking about the system. And finally, let's see. Yes, so one note on the callus for this system is that it tends to be a little bit more fragile. It feels like, it just can break very easily. But anyways, it's not like the most used type of tissue when it comes to propagating for disease-free crops. It tends to be used more for research and development purposes. And something that also caught my attention about these systems is that maybe because they're already working within, the safety cabinet and you remove the human factor for contamination, they're not really sealing the flasks. And usually, again, even at a small scale, we do seal the flasks with parafilm. So maybe it also has to do with the type of container they use. Finally, this one.
So what is ClickUp?
I always mute the ads. So this one just transplants the explants, the plantlets from one flask or container into the other. And I think the cutting is what's missing from their system, or at least as they show it here. And those are the ones I have for you guys. My questions, because since I didn't ask for quoting or anything, I don't know how much it would cost to make these. Again, it would work mainly for big companies. And I wonder how much sterilization can also, be automated in that sense, as in one of the first videos that I showed you, the rinsing in alcohol or chloride and so on. then yeah, based on what you saw, I wonder if it's really that complicated, because one of my main questions starting to write this piece was, why haven't I seen many of these systems yet? Is it because it's hard to work with plant tissues, like a very delicate grip? You know, is it because the computer vision data is not there yet? Can other systems be incorporated here? And media preparation, although I didn't put it in as, you know, within the section that requires this automation, it obviously tends to be a very repetitive process in any kind of biotech activity. So that would be great to have as well. I don't know if you have any more questions or yeah, anything else.
It's really interesting. I work with yeast more than plants, but yeah, it's kind of interesting to see how some of the ideas of automation can be applied to the plants. And I think that it's complex because of the different structures that plants can grow into, but these all show different ways to actually approach it. I worry that the chopsticks that the robot arms have would be maybe a bit too damaging when they go to pick them up, but clearly it seems to work.
Yeah, Scott, that issue was even when the person was doing it, like there was a point at which they were using tweezers and then they said to heck with it and just used fingers, you know, like, and so when would maybe soft robotics be more applicable than a tweezer kind of thing, you know, some of those kind of issues directly working with fragile roots.